Hello, and welcome to this Hawk Hydromet webinar on phytoplankton measurement using in vivo fluorescence technology. I'm Patrick Sanders, Southwest Territory Manager for Hawk Hydromet. Throughout our 30-minute webinar, I will give an introduction to phytoplankton, outline why phytoplankton measurements are important, review lab and field measurement methods, and provide an overview of the variables of in vivo fluorescence measurements. Phytoplankton are microscopic photosynthetic plants, bacteria, and autotrophic zooplankton that are commonly found in surface waters throughout the world. The word phytoplankton comes from the Greek phyton, meaning plant, and planktos, meaning wanderer. With these root word definitions in mind, it is important to note that phytoplankton are a type of free-floating algae versus a type that is attached to a surface, such as periphyton or benthic algae. Phytoplankton come in many different shapes and sizes and include green algae, cyanobacteria, also known as blue-green algae, diatoms, and dinoflagellates. Examples of what these different types of phytoplankton look like can be seen on this current slide. Also on this slide is an illustration of a couple of in vivo submersible fluorescence sensors that are measuring phytoplankton pigments directly in water. More details on these sensors will be discussed later in this presentation. Many phytoplankton appear green in color due to high relative concentrations of green chlorophyll pigments found within their cells. However, phytoplankton can also appear in other colors such as blue-green, red, brown, gold, yellow, and more if they have relatively higher concentrations of other pigments within their cells. Other common pigment types found in phytoplankton include the phycoprotein groups of pigments such as phycocyanin and phycoerythrin, as well as the carotenoid group of pigments, such as beta-carotene. Pictured on the slide is a satellite image of the South San Francisco Bay salt ponds in Northern California, as seen from Google Earth. The vivid colors that you see are due to high concentrations of phytoplankton, with the green ponds experiencing phytoplankton blooms rich in chlorophyll pigments, and the red shaded ponds experiencing blooms of phytoplankton rich with the pigment beta-carotene. The graph at the bottom right shows the light absorbance characteristics of common pigments found in phytoplankton, including the chlorophylls and beta-carotene. The pigments within plants and phytoplankton provide a vital function by absorbing the light energy used for photosynthesis. In this process, light energy from the sun drives a reaction where carbon dioxide and water are converted into sugars, with oxygen given off as a reaction byproduct. The actual photosynthesis reaction is illustrated on the bottom of the slide, while the graphics on the right illustrate the light harvesting process for green algae, shown in the top right, and cyanobacteria, shown on the bottom right. The light that powers the photosynthesis reaction in phytoplankton is harvested by various accessory pigments and then channeled to an energy-collecting pigment where a reaction center is located. This energy-collecting pigment is typically chlorophyll A, which is a pigment that is universally found in all phytoplankton. This universal presence, combined with chlorophyll A's natural fluorescent properties, makes this pigment an ideal and common measurement target for estimating phytoplankton biomass. Cyanobacteria also contain the chlorophyll A pigment, but they typically contain more of the accessory pigments named phycocyanin and phycoerythrin. As these accessory pigments are also naturally fluorescent, they are commonly measured for specifically estimating cyanobacteria biomass in water. Phycocyanin is commonly used for estimating cyanobacteria in freshwater systems, while phycoerythrin is commonly measured for estimating cyanobacteria in marine systems. There are many reasons as to why measuring phytoplankton is important as a water quality parameter. One example is for primary productivity quantification. As phytoplankton are able to create their own chemical energy through photosynthesis, they are commonly referred to as primary producers within aquatic ecosystems. These primary producers form the foundation of aquatic food webs, where they in turn are consumed by organisms higher up on the food chain, such as zooplankton, invertebrates, fish, and higher-level predators. The graphic on this slide shows a simplistic aquatic food web, where energy created by photosynthetic phytoplankton is channeled up through the food chain. As one might expect, concentrations of phytoplankton can have a direct effect on all organisms within an aquatic food web. Quantifying primary production through phytoplankton biomass measurements is a common way to gather direct insights into the baseline energy level of any given aquatic ecosystem. Another reason why phytoplankton biomass is measured is to gain insights on the level of eutrophication, or nutrient status, within an aquatic system. Eutrophication is the process where nutrients such as nitrogen and phosphorus are loaded into an aquatic system, typically caused by watershed runoff inputs. A eutrophic system is one where excessive nutrients have been added, and these systems are highly prone to phytoplankton blooms. An important consequence of phytoplankton blooms is that when phytoplankton cells die off and sink, their decomposition has the ability to significantly lower dissolved oxygen levels within a water body. This dissolved oxygen depletion can have direct impacts on the health of fish and other aquatic organisms. Phytoplankton monitoring can help water resource managers control watershed inputs that affect nutrient loading within an aquatic system. Certain species of phytoplankton, mainly cyanobacteria, can release toxins that can affect human and animal health. Some common cyanobacteria toxins include microcystis, which can affect the liver, anatoxin A, which can affect nerve synapses, cylindrospermopsin, which can affect the liver as well, and saxitoxins, which can affect nerve axons. Continuous proactive monitoring of harmful algae blooms as part of a broad management plan can help water resource managers to decrease prevalence of cyanotoxin-related health incidents. Continuous phytoplankton level monitoring can also be an important tool for monitoring drinking water reservoirs. Certain species of phytoplankton, mainly cyanobacteria, can produce compounds that can create taste and odor issues in water. A couple notable cyanobacteria byproducts that can cause significant taste and odor issues in water include 2-MIB and geosmin. In addition, phytoplankton blooms can cause drinking water reservoir filtration systems to work less efficiently. Continuous monitoring of phytoplankton levels can help guide proactive water reservoir management, such as by implementing algae control methods at the first sign of an impending bloom, or by providing data to help managers determine which source water intakes to use prior to filtration. I would now like to discuss common phytoplankton measurement methods, starting out with in vitro laboratory methods. Two of the most common types of phytoplankton measurement methods in the laboratory include cell counting methods and pigment analysis methods. Although laboratory methods can be time-intensive, carefully collected data from these laboratory methods provide highly valuable quantitative data in meaningful mass-to-volume units. Common units include cells per milliliter and micrograms per liter. Counting cells in a known volume of water is one common laboratory method of quantifying phytoplankton biomass. This method can be done either visually with a microscope or with instrumentation such as a flow cytometer. In the visual counting method pictured on the left, water samples are typically placed in a grid-lined counter cell, which is then placed on a microscope where individual cells can be manually counted by an observer. In the flow cytometry counting method, a flow cytometer instrument is used to count phytoplankton cells that pass through its measurement cell to determine cells per volume concentration. The graph shown on the right shows example data from a flow cytometer instrument. Other common laboratory methods involve various laboratory procedures, reagents, and instrumentation to isolate and measure specific phytoplankton pigments. These methods include in vitro fluorescence, spectrophotometry, and high-performance liquid chromatography, also known as HPLC. In these methods, specific photosynthetic pigments are extracted from the phytoplankton cells using various techniques, and then concentrations of these pigments are determined with analytical instrumentation based upon unique properties of these pigments. The three methods listed here each have their own EPA method associated with them for the measurement of phytoplankton. EPA method 445.0 is used for in vitro fluorescence, EPA method 446.0 is used for spectrophotometry, and EPA method 447.0 is used for high-performance liquid chromatography. Phytoplankton can not only be measured in vitro in the laboratory, but they can also be measured directly in the field using in vivo fluorescence sensors. In vitro is Latin for within the glass, a term that is used to describe measurement within the laboratory, while in vivo is Latin for within the living, a term used to describe field measurements of cells in their living state. In vivo fluorescence is a real-time measurement that generates instantaneous field estimates of phytoplankton biomass that can be directly correlated to quantitative measurements. This capability enables in vivo submersible fluorescence sensors to be used for any spot sampling, profiling, or continuous long-term unattended deployment applications.